Hi, I'm Soumya Pillai and welcome to another episode of Pure Science, where I dissect the biggest science story of the week. In the past, we have delved deep into the science of gene editing, the technology, the advancements, and how it is being hailed as one of the greatest medical breakthroughs of the 21st century. And for good reason, because for decades, doctors could only treat the symptoms of many inherited diseases. But gene editing promised something revolutionary. It helped doctors and researchers not just treat diseases, but correct the faulty DNA that causes them in the first place. We have also seen that scientists have even managed to develop personalized treatments designed specifically for a single patient carrying a unique genetic mutation with the help of this technology. But this week, we are venturing into a completely different side of gene editing. An investigation carried out by Science magazine this week has cast a spotlight on the risks that come with this new era of medicine. Not because gene editing itself has failed, but because of allegations that a patient's death was never fully disclosed to the scientific community. The story raises an important question. How much transparency is needed when scientists are testing some of the world's most powerful medical technologies? So let us first understand what happened. The investigation centers on a 6-year-old girl in China who suffered from an extremely rare genetic mutation in a gene called MECP2. While the exact disease the patient suffered from is not known, it is said that the child had experienced a seemingly normal development for the first 1 to 2 years of her life, but then began to lose previously acquired skills. She had started speaking in simple sentences, was slowly losing the ability to walk and use her limbs properly for everyday movements, along with facing a risk of other severe developmental disabilities. And unfortunately, there is currently no cure of this disease. Because the girl's mutation was unique, researchers decided to build a customized treatment specifically for her. This is part of a rapidly growing field called personalized gene editing, sometimes referred to as N of 1 medicine. Instead of creating one drug for millions of people, scientists design a therapy for just one individual. It sounds extraordinary and it is, but it also means there is very little previous experience to guide doctors on whether such treatments are safe. To understand the treatment, we first need to understand how gene editing works. Our DNA is made up of billions of chemical letters A, T, C, and G. Every gene is essentially a long sequence of these letters. Sometimes changing just one letter is enough to cause a devastating disease. Just a side note, in the 6-year-old's case, there was a mix-up in the T and the C letters. Now, most people have heard of CRISPR, often described as molecular scissors. Traditionally, CRISPR works by cutting both strands of DNA at a specific location. The cell then repairs the cut, allowing scientists to disable or replace a faulty gene. But the treatment used in this case employed a new technology called base editing. Base editing is often described as a molecular pencil rather than a molecular scissor. Instead of cutting the DNA completely, it changes one DNA letter directly into another. This makes it much more precise and, in theory, safer because it avoids creating double-stranded breaks in the DNA that can sometimes lead to unintended mutations. Scientists consider base editing one of the most exciting advances in genetics because many inherited diseases are caused by a single spelling mistake in the DNA. If you can simply correct that one letter, you may be able to cure the disease. The researchers developed a customized base editing treatment and delivered it directly to the girl's spinal fluid, allowing it to reach cells in the brain and the nervous system. Because the therapy had to enter human cells, it was packaged inside an engineered virus called an adeno-associated virus or AAV. AAVs are commonly used as delivery vehicles in gene therapy. They are modified so they cannot cause disease but can transport genetic material into the cell. In simpler terms, this is like a microscopic courier service carrying the gene editing machinery to the right destination. However, these viral delivery systems also come with risks. If the immune system recognizes the virus as foreign, it can launch a powerful inflammatory response. In recent years, scientists have become increasingly aware that very high doses of AAV-based therapies can trigger severe immune reactions, liver damage, and in other cases, even death. According to the science investigation, this is exactly what appears to have happened in the case of the 6-year-old patient. Within days of receiving the experimental treatment, the child developed a severe immune response. Despite intensive medical care, she died. Now, experimental treatments do carry risks. Every early stage clinical trial involves uncertainty, and unfortunately, not every patient survives. That by itself is not what has shocked the scientific community. The real controversy is what happened afterwards. According to the investigation, the girl's death was never reported in any public clinical trial registry. Researchers later published a paper in the journal Nature describing the successful development and testing of the customized base editor in laboratory and animal studies. But the paper reportedly did not mention that the treatment had already been administered to a child, nor did it mention that the child had died following the treatment. The investigation also reports that the girl's family had contributed roughly around 800,000 US dollars towards the development of the therapy, highlighting another emerging feature of highly personalized medicine, which is its enormous cost. Now, why does all of this matter? Because science only advances when researchers report both successes and failures. Imagine if aircraft manufacturers only reported successful flights and quietly ignored crashes. Engineers would never know what actually went wrong. Regulators couldn't improve safety standards, and passengers would continue facing avoidable risks. Medical research works the same way. Every unexpected complication teaches scientists something valuable. If a patient develops a severe immune reaction, future researchers need to know why. Was the viral dose too high? Was the immune system already primed? Was the delivery method unsuitable? Could the same complication happen again? Without that information, other researchers may unknowingly repeat the same mistakes. Several bioethicists interviewed for the investigation argued that omissions of such a significant clinical event undermines scientific transparency. Some have questioned whether the published paper should be corrected to include the full context surrounding the therapy's development. The investigation has also drawn attention to how experimental gene editing therapies are regulated. Unlike conventional medicines that undergo large clinical trials involving hundreds or thousands of patients, personalized therapies often involve only a single individual. That creates a difficult challenge for regulators. Traditional drug approval systems were never designed for medicines that may only ever be used once. Around the world, scientists and regulators are still trying to develop rules that balance two competing priorities. On one hand, patients with fatal rare diseases often cannot wait years for conventional clinical trials. On the other hand, speeding up the access cannot come at the cost of patient safety or scientific accountability. Now, it is extremely important to emphasize that this case should not be interpreted as evidence that Gene editing itself is unsafe. In fact, gene editing therapies have already transformed the lives of many patients. The first CRISPR-based treatments approved for sickle cell disease have shown remarkable success, and dozens of clinical trials are currently underway for inherited blindness, blood disorders, immune diseases, and certain forms of cancer. The technology remains enormously promising. But every powerful technology requires equally strong safeguards. While on this topic, many people might remember another episode from China in 2018, where the scientist He Jiankui announced the birth of the world's first gene-edited babies. The experiment was condemned internationally not because gene editing lacked potential, but because ethical standards and transparency had been ignored. This latest investigation reminds us that those lessons remain just as relevant today. The future of medicine is undoubtedly moving towards personalized treatments where therapies are tailored to an individual's DNA. That future has the potential to save countless lives. But innovation alone is not enough. That was all from me this week. I am Soumya Pillai and this was Pure Science.